In my last video, I covered all of the information you were required to know for Chapter 8 photosynthesis. The next step we're going to take in understanding cellular energetics is the breakdown of the molecules that are produced in photosynthesis and releasing the available energy and the chemical bonds available. So the objectives for Chapter 9, cell respiration, is first, be able to define cellular respiration. This may also include understanding the chemical equation involved, the chemical balances in terms of how many molecules of each reactant are present, what are the products, and how much of those molecules are available. So I'll take you step by step through that. We're also going to highlight the major events that occur during glycolysis, so all of the reactants, all of the products, and where it takes place. We're also going to compare and contrast lactic acid fermentation and alcoholic fermentation. So those are two alternative pathways that certain cells can use to continue to process the pyruvate into a temporary storage molecule. And we'll explain kind of how they're different and how they're similar as well. And the last thing we'll do is calculate the efficiency of glycolysis. So we'll look at the efficiencies of that process, how much ATP is generated, what the products can soon become, how much available energy is there to be used, as well as the alternative pathways. So first, as just an introduction, think back to Chapter 8, where autotrophs are making energy. In the case of cellular respiration, it's the harnessing and the use of that made chemical energy. So, our cells need energy for all the activities they must follow throughout the day. So the organic compounds, things like carbohydrates, lipids, and proteins that are made by plants, can be converted and then transformed into usable energy. So the organic compounds are coming from photosynthesis. So in the case of photoautotrophs, they use that process to make the organic compounds. And when cells break down organic compounds into simpler substances, it releases energy, or what we call ATP, adenosine triphosphate. So cellular respiration is not a simple process. There are many steps, many stages, things that you have to understand. But the basic premise is organic compounds are going to be broken down and made available into readily usable energy, or ATP. So biochemical pathway that is the beginning of cell respiration is glucose. The breakdown of glucose, I should say. So glycolysis is the biochemical pathway that is the beginning of cell respiration. It does not make a lot of ATP. It's very fast, it's very easy, it's not complicated, it's short, it's brief, it's not that efficient, but it is quick. So obviously it yields a small amount of ATP, but there are some other things that can occur after that will produce larger amounts of ATP. Now the two main pathways are aerobic respiration, which is if oxygen is present, and then there's anaerobic respiration if oxygen is lacking. So we have aerobic respiration in the case of cellular respiration, and then if oxygen is lacking, we call that fermentation, which is an anaerobic process. Remember, anaerobic conditions don't require oxygen, so there's no O2 required. Fermentation can proceed in certain cells. Under aerobic conditions, that means oxygen is prevalent. There's probably large amounts of it. ATP can be synthesized using the oxygen as a very important reactant. We'll talk about the Krebs cycle and the electron transport chain and how they're important usages of the oxygen that's available. So let's start with the first step. All cells undergo glycolysis. So the starting molecule is glucose, containing 6 carbons, which becomes oxidized during glycolysis, producing 2 3-carbon molecules, which we'll refer to as either pyruvic acid or pyruvates. Now here's the thing you must understand. Glycolysis is technically anaerobic, does not require oxygen to take place. Therefore, this occurs in the cytoplasm or the cytosol of the cell. So in the grand scheme of things, big picture, imagine a cell with its cytoplasm. There is constantly glycolysis occurring in the cytoplasm. So the 6-carbon glucose being converted into 2 pyruvates or pyruvic acids, which are 3 carbon each. The net gain of this process is 2 ATPs. It costs an investment of 2 ATP. It generates 4 ATP, so there's a net gain of 2. It also produces the 2 pyruvates, and it also produces 2 NADHs, which is an electron carrier that's going to be important in the electron transport chain later. So some things to keep in mind. Glycolysis is by definition anaerobic. It does not require oxygen to take place. It takes place within the cytosol or cytoplasm of the cell. It does have an initial investment of 2 ATPs, produces 4 ATPs, so a total net gain of 2 ATP. It's fast, it's easy, and it's constantly happening within the cytosol. It's not hard for the cell to do this. It is not technically, efficient, but it does offer the ability of the cell to create some amounts of ATP. So here's a step-by-step process. Step 1, you have glucose, the 6-carbon molecule. You see the 2 ATPs are invested. That energy is going to be used to split the glucose molecule into 2 3-carbon molecules, ending up as pyruvates. Now in the process, 2 NADHs have been generated, 2 pyruvates have been generated, and 4 ATPs have been generated for a net gain of 2 ATP. So those are your products. Here's another way to look at it. Pause the video if you'd like to take a look at a step-by-step breakdown of what occurs during glycolysis there. Now, if oxygen is not present, after glycolysis, some cells can undergo fermentation as an alternative pathway. Now, this is anaerobic. There's no oxygen required. Some cells can turn that pyruvic acid or pyruvate into other compounds. Now, since it is anaerobic, it is going to occur in the cytosol or the cytoplasm. It does not technically produce ATP. It produces a couple of different compounds that initially won't produce ATP, but it temporarily converts them to be reused later for something else. But it does regenerate NAD+, which can be used to continue glycolysis. So it does create more of the electron carriers, converting into NADH later. Now, the type of fermentation that occurs is going to differ from cell to cell. Every cell is going to use a different set of enzymes. Every cell may produce a different type of compound byproduct. So the 2 major examples of fermentation are lactic acid fermentation, which takes place in the pickling process with jars of pickles. It takes place in human muscle cells. Those are examples of it. And then there's alcohol fermentation type 2, which is like, you think about yeast cells that are used in baking, and you think about the yeast that is used in the alcohol industry and how they make alcohol for beverages, champagne, wine, beer, and so on. So we'll talk about those 2. So let's first start with lactic acid fermentation. So there's unique enzymes that can actually create lactic acid fermentation following glycolysis with all that additional pyruvate. Now, these are microorganisms that ferment and produce different types of foods for us. So the dairy industry, like with yogurt and cheese, also in our human muscle cells, animal cells, not just humans, but all animals, their muscle cells are able to also undergo lactic acid fermentation during very strenuous activity. So you think about the times when you're sprinting or you're doing any type of muscle training, weight training. Those last few repetitions are very, very tough. You feel burning in your muscles, a burning sensation. That is an indication of lactic acid fermentation occurring. So here's the pathway. You start with your 6-carbon glucose that undergoes glycolysis. You have your pyruvates are made, and then a shift because there is an absence of oxygen. Instead of the pyruvic acid going into the Krebs cycle in the electron transport chain, if oxygen is there, oxygen is not there. So instead of going that way with oxygen, it gets reverted into lactic acid as a secondary byproduct of breaking down the pyruvate. Now remember, this is anaerobic, so it does take place in the cytoplasm of cells. So again, I gave the example of human muscle cells. It's a situation where muscle cells are actually running out of oxygen and they're using it all and it's becoming depleted. So they shift from using aerobic processes to an anaerobic process like lactic acid fermentation. And lactic acid, being an acid, it's very low pH, so it creates this burning sensation, creates muscle fatigue. You start to get cramps, you may feel pain, it may cause your muscles to even fail. They probably either ran out of glucose, couldn't process it fast enough, or in the case of most cases, ran out of oxygen. The oxygen depleted was used completely, and now it's left to only ferment using lactic acid fermentation. The other process or pathway is alcoholic fermentation. So this is the process by which unicellular organisms and some plants can actually break down pyruvate into alcohol or ethyl alcohol, sometimes called ethanol. The enzymes used to convert pyruvic acid into ethyl alcohol are unique, and they have a byproduct. It actually creates CO2 gas as a byproduct. This is the basis of the beer and wine industry. So alcoholic beverages, that contain ethyl alcohol or ethanol, use this process, use these microorganisms to break down the sugars into alcohol. So here's the process. You start with glucose on the top, breaks down through glycolysis. You now have your pyruvic acids. The next process that occurs is the enzymes are going to start to break down the pyruvic acid, or the pyruvates, into a 2-carbon compound called ethyl alcohol. And as a byproduct, it also produces carbon dioxide. So the way that you would know that yeast is fermenting in dough is you'll see that the dough is starting to rise. You'll see bubbles forming. You'll see CO2 gas escaping from the dough. And you'll smell the alcohol has been produced. So it's something that you can definitely observe and identify. So the baking industry, along with the adult beverage industry, also utilizes this process. The CO2 is definitely visible and observable. You'll see the bubbles in the dough. And obviously, this is why we have to bake dough. To make bread, you have to cook all the ethyl alcohol out of it. Now, glycolysis, going back to it, the first step is not very efficient. It's only about 3.5% efficient. Remember what I said earlier. It's fast, it's easy, but it's definitely not efficient. Not much ATP is made. So there is a trade-off. The positive thing is you get a little bit of energy very quickly. The negative is it's not efficient and it doesn't produce a lot of ATP. Anaerobic pathways generally are not efficient. So the same can be said about fermentation with respect to the lactic acid process or with respect to the alcoholic fermentation process. But, in the case of smaller organisms like yeast, which are microorganisms, or bacteria, they don't need a lot of energy. So maybe those anaerobic pathways in glycolysis and fermentation, maybe that's enough. Maybe they only need a little bit of ATP at any given moment. But when it comes to making large amounts of ATP, oxygen is going to be required. This talks a little bit about the evolution of respiration pathways. So here's the overview of aerobic respiration. This is when oxygen is available. It's 20 times more efficient as glycolysis by itself. So if glycolysis makes, a net gain of 2 ATP, this is going to be 20 to 25 times more efficient. So 2 major stages of aerobic respiration are the Krebs cycle and electron transport chain. These are going to occur after glycolysis if oxygen is available. The first stage is the Krebs cycle. This is where the pyruvates from glycolysis are going to be processed. What's kind of interesting here is there's not much ATP made here either. So remember, there was a net gain of 2 ATP in glycolysis. There's also a net gain of 2 ATP in the Krebs cycle. But there are tons of electron carriers produced here. We'll talk about what happens there. The Krebs cycle does take place in the mitochondrial matrix, which again, the mitochondria is like a cell within a cell. That means that it has a double membrane, it has its own unique DNA, just like the chloroplast. It has, like I said, a double membrane, outer and inner. The outer membrane is going to allow the pyruvic acid from glycolysis to diffuse across, enter the mitochondrial matrix, which is the fluid of the mitochondria, and that's where the Krebs cycle takes place. Then the last stage is the electron transport chain. This is where all the electron carriers, NADH, FADH2 from the Krebs cycle are going to be used. This is where most of the ATP is made. The location of all aerobic respiration, the Krebs cycle, the electron transport chain, that's going to take place in the mitochondria. So here's the structure of the mitochondria. You can see it's a bean-shaped organelle. It is the ATP synthesis location of the cell, the eukaryotic cell. So you can see the matrix, which is the fluid inside of the mitochondria. The cristae is the inner membrane, kind of the inner linings, the inner foldings, and then the outer membrane on the outside. So the first statement there says, in the Krebs cycle, there are 2 turns. The reason there are 2 turns is there are 2 pyruvates per glucose from glycolysis. So each turn produces 4 NADH, but there's 2 pyruvates or 2 turns. So there's 8 NADH electron carriers. Those go to the electron transport chain. There's also 1 FADH2 per pyruvate, but there's 2 pyruvates per glucose, so there's 2 FADH2s. Then there's 1 ATP made per turn of the Krebs cycle or per pyruvate, but there's 2 pyruvates per glucose, so that's 2 ATPs. There's also a waste product in the Krebs cycle, and it has to do with the oxidation of the pyruvates. When oxygen combines with the 3-carbon pyruvate, it snatches up a carbon from the pyruvate, creating CO2. CO2 is a byproduct that is going to diffuse out of the mitochondria and out of the cell. So per pyruvate, per turn of the Krebs cycle, 3 CO2s are processed. But there's 2 pyruvates per glucose, so there's 2 turns. That means there are 6 carbon dioxides produced. Definitely go back over this to see what is produced. The last step of aerobic respiration is the electron transport chain. This takes place in the cristae, or the inner membrane of the mitochondria, in eukaryotic cells. This is where a large portion of the ATP is generated from the electron carriers from the Krebs cycle, as well as glycolysis. So the NADHs, the FADH2s, have a fate. They're going to enter the electron transport chain and get generated into ATP. So the way this works is, this looks strikingly similar to the inner membrane of the thylakoids where the photosystems reside, right? The electron transport chain there. You've got clusters and groupings of different proteins and basically they're cell receptors, right? They're going to be recharging and combining to make all this ATP in the electron transport chain. So you can see where the H plus ions are. You could see that they're going to be used here in this case. Those H plus ions reside with the NADH and the FADH2s. Now the final electron acceptor in the electron transport chain is oxygen. Remember, oxygen was used in the Krebs cycle to help oxidize the 3-carbon pyruvates, and it created CO2. Oxygen is also used in the electron transport chain. It's the final electron acceptor. So what forms when you get oxygen and hydrogens? You get H2O. The final molecule that is formed in the electron transport chain in aerobic respiration is water. So the hydrogens combine with the oxygen that's available in the mitochondrial matrix and also in the electron transport chain. We now have water formation. You can't get any ATP formation unless water is formed. Oxygen is the final electron acceptor. So now we understand the importance or the role of oxygen. It is the final electron acceptor. The H plus ions supplied by the electron carriers form the water molecule. So you'll see in the equation you have oxygen, you have the electrons, and you have the hydrogen, positively charged hydrogen ions that form the water molecule. So here's a good view of what happens. You can see the process starting on the left. You have the NADHs make the hydrogens available for use. You have the FADH2s, they make their hydrogens available for use to form and combine with the oxygens to form H2O. You can pause the video to take a look. So here's an overview of what takes place during cellular respiration. Starting on the left, you have glycolysis, glucose being converted into pyruvate, a 6-carbon molecule converted into 2 3-carbon molecules. There are some NADHs made, there are 2. There is some expenditure of ATP, cost 2, but it does produce 4, which is a net gain of 2 ATP. The pyruvates enter the mitochondria, they cross the outer membrane, entering the mitochondrial matrix to start the Krebs cycle. Now this is if oxygen is present. So then the oxygen is used to oxidize the 3-carbon pyruvates into citric acid and a couple of other things. It does form 2 ATP. It does form 8 NADH molecules. It does form 2 FADH2 molecules, and it forms 6 carbon dioxides. Now keep in mind there are 2 ATPs made in Krebs cycle. So if you're counting, net gain of 2 in glycolysis and also a gain of 2 ATP in the Krebs cycle. So far that's only 4 ATP. Now the electron carriers from the Krebs cycle then go to the inner mitochondrial membrane, or the cristae, to be oxidized. This is where a lot of your energy is going to be made. So you've got 10 NADH2, you have 10 NADHs, 2 from glycolysis, 8 from the Krebs cycle. You have 2 FADH2s from the Krebs cycle. Those are going to be oxidized. Remember, the H plus ions are used to form water. Oxygen is the final electron acceptor. Each NADH molecule is worth 3 ATP. You have 10. That's 30 ATP. Each FADH2 can form 2 ATP molecules. So that means 4 ATP. So the electron transport chain can make up to 34 ATP. The Krebs cycle makes 2. The glycolysis makes 2. Well, it gains 2. For a grand total of theoretical yield of 38 ATP from this entire process. So there's the math. You definitely want to go back and revisit this. Take a look at each step, what the products are. Take a look at each step, what ATP molecules are synthesized, what byproducts there are, and the way things kind of happen. There's another visual to help you kind of understand how it's made. Now, 38 ATP from one glucose molecule may seem like that's a little bit high, and it kind of is. That's the theoretical yield, but there's a lot of factors that can prevent one glucose molecule from turning into 38 ATP. It's probably more realistic that would be closer to 29 or 30 ATP. So here's a little bit about the efficiency of aerobic respiration. If you remember from the glycolysis slide previously, glycolysis is only about a 3.5% efficiency. Aerobic respiration is much more efficient, probably closer to 60 or 70% efficient. So your cells would prefer to go through aerobic respiration because they can make much more ATP. And if you're a large organism, you want more ATP per glucose. Glycolysis is just not enough. So it talks a little bit more about that there. Now, this is a good table to go over where things are taking place, what things are happening, what the products are, what the reactants are, and so on. Definitely take a look at that. This is kind of an interdependent slide. This talks about the importance that photoautotrophs play and how all organisms kind of rely on photosynthesis to make energy in terms of carbohydrates, and then cells breaking down that available chemical energy in those molecules in cell respiration. So do tie in the ideas that the chloroplast is responsible for synthesizing carbohydrates, and then the mitochondria is the organelle that is important in aerobic respiration, breaking down the glucose into usable forms of energy. Also keep in mind that chloroplasts are involved in making or synthesizing the O2. So if a mitochondria is going to undergo cell respiration, it needs oxygen to efficiently break down glucose. So it's a cycle. Definitely go over the similarity slide so you can look at the energy carriers, the electron carriers, the reactions that take place, the concentration gradients, the biochemical pathways. This is a really important slide to go over. I'll let you look at that. And then here's your similarities and differences part. So what's different about photosynthesis and cell respiration? What I would urge and encourage you to do is look at these questions and see if you can answer them. And then re-watch parts of the video to make sure that you understand. These are actually questions that will go with our lab that we're going to do. So I'll be posting this slide presentation for you guys to answer. These are your post-lab questions. They'll make more sense once you finish the lab, make your observations, and collect your data. So that's it for the cell respiration video. Thank you guys for watching. Remember, at any point you want to pause the video, rewind, rewatch, relearn. Just go back to the part and go over those things. Take a look at all the moving parts. It's a very dynamic process. Just keep in mind that this is the process that cells use to free up energy in carbohydrates and other macromolecules. So do keep that in mind. And again, rewatch, pause, rewind, take notes so that you can learn it as best as you can. I hope this helps you guys a lot. Let me know if you have any questions, just ask in class, and I'll be there to help you guys.